Okay. So hello, everyone. I guess that we already started recording, so let's wait a few seconds to let other people get in. I can see that the numbers are increasing, so just 1 or 2 minutes for other people to join us, and we will start. Okay. I can see some faces that I know, some completely new people. So when we are waiting for the rest, maybe you can use the chat and write where you are from which country. So are you a trainer or are you an instructor or maybe a researcher? It could be nice if you tell us a little bit more in the chat, just for 2-3 minutes, and we will start. Okay, I will share the screen in the meantime to start the presentation. Okay, thank you. England, Poland, I can see some of my students, and also England. This one is, let me check, Scientific Advisory Board Member, Active Pregnancy Foundation, perfectly. Italy, very nice. Romania, who else? Group fitness instructor in Romania as well. Canada, South Africa. So very nice that we can see different representations. Roya from Poland, Montreal, Canada. Okay, so that's really nice. So I will start sharing the screen and still very slowly letting other people join. Okay. Please confirm that you can see the screen, my presentation. Yes? Okay, perfectly. So I think that we can start. So officially, good morning, good afternoon, and good evening, everyone. I'm very, very happy to chair this webinar today. Pregnancy exercise today for a healthier world tomorrow. This is our title. And I have eminent guests. My name is Anna Szumilewicz. I'm from Gdańsk University of Physical Education and Sport in Poland. And my guests are from Northern America. And we have Professor Michelle Motola from Canada. Hello, Michelle. We have Professor Laurie Goodyear from US. And hello, Laurie. Hello, yes. And we have Professor Linda May from US as well. So this is morning for them. For us, I guess we have nice afternoon, and I could see some people from China. So this is probably evening, not very late, but still evening. So I'm very happy that you are connecting. And before we start, I will start to answering the most frequently asked question. Yes, this webinar is being recorded and we will post it on the website. So you have the QR code. Now, when you use the QR code, you can see only the information, general information, of course, but we will post the video material there. Just give us a couple of days and it will be ready for you. But I hope that those people who are with us now, that you will stay with us by the end of the webinar and that you will get all the necessary information that you need. I just would like to announce, to inform you that this webinar is a part of a series of webinars. First, we started in February with the webinar organized mostly by European College of Sports Science. Now, European College of Sports Science is also our partner, but we co-organize it with American College of Sports Medicine and with other institutions that support physical activity generally, not only during pregnancy and postpartum, but generally for the whole population of human. So these are the webinars that I recommend for you. Maybe in your native languages, it will be easier. I know I'm not a native speaker, so I understand that sometimes we are struggling with understanding what's going on. So this is an option for you: Spanish version, a Portuguese version, Lithuanian, and Polish. So I suggest to go to our website and use the recordings. These webinars were really very successful. And this is scheduled for today. We have, as I mentioned, four presentations. First, we will start with Professor Michelle Motola on impact of exercise during pregnancy on chronic disease risk. Then Professor Laurie Goodyear, effects of maternal exercise on offspring health, epigenetic mechanisms. Professor Linda May, maternal exercise influence on infant myogenic and adipogenic response, and my presentation about high intensity interval training. And I will be talking about how we can improve the conditions for fetal development. But the main goal at the end will be not my research, but my offer, not my, but our offer to promote physical activity globally. And I would like to invite you to the Global Active Mother Day on May 11. I will talk about it at the end of the webinar. So Michelle, you can start sharing the screen now, and before you start, just a few words about Michelle. I know that those people who are interested in physical activity during pregnancy and postpartum know Professor Michelle Motola, but just a few words from my side. So she's the professor at the University of Western Ontario, London, Canada. And she also is the chair of Maternal Fetal Newborn Health Division. And she's involved in Children's Health Research Institute, Board of Trustee member. She's the past chair of the Special Interest Group for Pregnancy and Postpartum at the American College of Sports Medicine and director of Samuel McLaughlin Foundation. I'm not sure if I'm pronouncing it correctly. Exercise and Pregnancy Lab. So probably it's very funny for you, but I'm struggling, but I'm trying to pronounce it correctly. But also I would like to emphasize that Michelle was one of the first professors who was dealing with pregnancy exercise many years ago. And actually, I think that you started this trend, and now we have more and more studies in this topic, but the beginnings were not very easy. So thank you very much for your all achievements, and I'm very proud and honored that you are here today with us. So Michelle, the floor is yours. Start your presentation, please.
Thank you very much. Can you see my screen?
Yes.
Perfect. Okay, welcome everyone, and thank you very much, Anna, for inviting me today. I'm going to be talking to you about the impact of exercise during pregnancy on chronic disease risk. And my objectives today are to define factors during pregnancy that influence future disease risk in the infant in particular. And this is based on the theory of developmental origins of health and disease, or DOHAT. I'm also going to talk about examining how exercise during pregnancy can mitigate future disease risk in both the mother and the infant. The way I like to start is to introduce you to the topic with regards to saying that the egg that formed you was created when your grandmother was pregnant with your mom. And so what that means here is that this individual is your grandmother. She's the mother of the first generation who is pregnant with your mother here, which is the fetus, the second generation, and that little egg cell or ova that is developing within that fetus is going to become you. And so what that means is that the maternal environment is extremely important, not only for the mother, the fetus, but also for 2 and 3 generations in the future. And this is basically called epigenetics or fetal programming. It's a very complex area, but that's very simply just saying that the maternal environment is extremely important for future chronic disease risk. And this developmental origins of health and disease is a theory, or also called Barker hypothesis, that talks about the adverse influences early in development, especially in utero, and this can result in permanent changes in physiology and metabolism, resulting in increased risk of disease in adulthood. And this can be potentially bad events where the maternal environment can be potentially having risk factors for the fetus later in life, or like, for example, maternal diabetes or hypertension, or it could be good events. So speaking specifically about setting the stage for a healthy lifestyle. And when you think about risk factors, one of the things that I think that really encompasses everything that we talk about with regards to risk factors for chronic disease is physical inactivity. And this can definitely be the driving factor potentially for obesity, excessive gestational weight gain, or gestational diabetes mellitus, which is diabetes first diagnosed during pregnancy. And all of these potential issues with physical inactivity puts the mother and the baby at risk for type 2 diabetes, cardiovascular disease, and metabolic syndrome. And let me talk a little bit about excessive pregnancy weight gain. And this is from the Institute of Medicine that has weight gain guidelines for pregnancy. Over here, you can see that there are pre-pregnancy body mass index, here, BMI, and giving a range of total weight gain for individuals. So for example, anybody that is normal weight, they should not gain any more than 35 pounds or 16 kilograms. And you can see that as the BMI increases, so the amount of weight that the individual should gain decreases. And they also suggest rates of weight gain for second and third trimester. And again, that individual with normal weight should not gain any more than 1 pound or 0.5 kilograms in second and third trimester. And this assumes that the individual gains only 4 pounds or 2 kilograms in the first trimester. So what can happen then if we can maybe mitigate this, and instead of having physical inactivity, we now have physical activity? And what that can potentially do with a modifiable risk factor is we can prevent excessive gestational weight gain, and now we have adequate gestational weight gain that we can then prevent obesity, prevent gestational diabetes. And what that means is that we could then also prevent chronic diseases in the mother and the downstream effects on the future child. Now, we were lucky to receive funding from the Canadian Institute for Health Research, and we did 3.5 years worth of work where we brought together a consensus guideline, consensus panel made up of experts in the field, methodologists, and stakeholders, where we basically wanted to take a look at the literature. And this literature, we looked at, screened almost 27,000 titles and abstracts, and we had 675 studies that were included, and we ended up with publishing 12 systematic reviews on the impact of maternal exercise. And these are published in the British Journal of Sports Medicine. And we also published the 2019 Canadian Guideline for Physical Activity throughout pregnancy. And basically what these 12 systematic reviews tell us is that physical activity reduces the risk of gestational diabetes by 38%, gestational hypertension by 39%, preeclampsia by 41%, excessive pregnancy weight gain by 32%, depression by 67%, urinary incontinence during pregnancy by 51%, and in the postpartum period by 47%. And we can also decrease those big babies or macrosomic babies by 39%, and also prevent instrumental delivery by 24%. And what we also found was that pregnancy is not associated with miscarriage, stillbirth, or neonatal death, birth defects, preterm birth, low birth weight, neonatal hypoglycemia, or birth complications. And we also found that it's not associated with changes in fetal heart rate or uteroplacental blood flow metrics. And what this potentially means is that what we've got now and what we should be promoting is that physical activity represents a foundational shift in our view based on what we see. And now we're going from recommended behavior to improve quality of life to now a specific prescription for physical activity to reduce pregnancy complications and to optimize health for 2 and 3 generations. And so it's extremely important that we implement physical activity into clinical practice for lifelong benefits for both the mother and the child. And what I'd like to do here is to just share with you this baseline here. So as the quantity and quality of maternal exercise increases, we see a maternal dose response curve where we see that there's a decreased risk of gestational diabetes and hypertension. There's a promotion of normal glucose tolerance and psychological benefits in this maternal dose response curve. But we can also see that there's a fetal dose response curve where in this area we see beneficial fetal and placental adaptations that occur as the quantity and quality of maternal exercise increase. However, we do know that there's an optimal zone for maternal exercise prescription, and that's based on the literature and what our Canadian guidelines are also based on. But the potential issue here is that we don't know where that upper threshold limit is. And this is where we need more information and we need more guidance and evidence in this upper threshold limit because we don't know where we might start seeing potential issues or problems. But I'd like to have your attention focus here on the other end of the spectrum, where now I believe personally that pregnant individuals are not getting enough physical activity to even make it to this baseline level. And what we now are seeing is altered maternal pregnancy adaptation. So the mother is now having gestational diabetes or hypertension during pregnancy. And we're also seeing altered fetal development. So now we're starting to see bigger babies or macrosomic babies and babies that are born too small. So I think that we need to be very cautious of all of these factors. But one of the things that I think is extremely important during pregnancy is that it's a time when many individuals change to a healthier lifestyle. They improve their eating habits, they quit smoking, they stop alcohol use, they maybe moderate their caffeine consumption, but they also might be thinking about active living. And this is where I think we can make a difference. And physical activity does not need to be in a supervised setting or with specific equipment. In fact, activities such as walking can have positive benefits because walking is actually an aerobic activity. And we need to think about motivating our potential pregnant clients and have them accountable so we can have face-to-face phone calls, pedometers, technological tools so that they can wear on their wrist or download phone apps, text messaging, other tools that you can think of that maybe enhance and promote healthy habits. And when we think about the guidelines that are out there for physical activity, we're talking about 150 minutes per week, which is usually about 30 minutes in 5 days. But I want you to think about what happens during the day and the other 23 and a half hours that your potential client can maybe think about being physically active. And I just want to promote here this YouTube video by Dr. Mike Evans called Let's Make Our Day Harder. And it's a great way to introduce how to promote increases in daily physical activity through things like taking the stairs or parking farther away. And healthy lifestyle is a balance, and I'm sure you all know this, but what we suggest here is when we're thinking about eating habits, we talk to our pregnant clients, get them to not eat for 2, but eat twice as healthy and to watch their portion sizes. And of course, physical activity is also extremely important because we want to promote physical activity during pregnancy to prevent chronic disease risk in both the mother and the developing fetus. So our job, I believe, is to grow healthy babies. And I borrowed this from Ann Geddes because I think that's exactly what we're doing here. We need to make a difference for 2 and 3 generations. And I want to take a couple minutes here to promote our 2-day mini conference that's going to be happening at Western University in London, Canada. And it's a 2-day mini conference, and the title is Health Behaviors in Pregnancy and Childhood. And it's going to be on Tuesday, October 14th and Wednesday, October 15th. And it is the pre-conference to the Canadian Society for Exercise Physiology. And the pre-conference is for free. And basically, this is what we've got planned for the Tuesday. We're going to look at cultural competence and humility. We're going to look at metabolic destiny before birth. We're going to look at the impact of physical activity during pregnancy and future chronic disease risk, maternal exercise on the placenta, how we can hit maternal metabolism through HIIT to enhance neonatal outcomes, and the impact of structured exercise on infant health, and the impact of pregnancy using diet and exercise on offspring neurodevelopment and early childhood. So the morning sessions will be regarding pregnancy, and the afternoon session will be looking at movement behaviors of children and adolescents, and that's the Tuesday. And then we have a special thing happening in the evening with a special community event where we will be releasing the CSEP sponsored 2025 Canadian guideline for physical activity, sedentary behavior, and sleep throughout the first year postpartum. And the Wednesday, also the pre-conference, from 10 till noon, will be a symposium looking at the historical fallacies and misinformation regarding exercise during and after pregnancy and how current evidence clashes with these ideas. We've got 4 speakers. And then in the afternoon, we're going to do another symposium that's going to look at the 24-hour movement guidelines for the early years and children and youth. So please stay tuned. This is the first announcement that we've made for this for the fall. We will be having abstract deadlines, and what we want to focus on are trainees. So this will give the trainees the opportunity to deal with and interact with experts in the field. And we're going to be asking for abstracts so that our trainees can provide posters, and there will be awards for the best posters for the trainees. So I'm going to leave it there and say thank you very much for your attention, and I would be more than happy to answer any questions.
Yes, thank you very much. Thank you very much, Michelle. So do we have any questions from the audience? Would you like to ask something? Usually the participants are rather shy. So let's wait. If you don't have the question now, remember we will have time at the end of this webinar as well. So maybe waiting for your question, I will have a question to Michelle. Michelle, could you tell us what is, or maybe what are the biggest changes in the approach to pregnancy exercise that you can observe during these years that you are involved in many pregnancy projects? So can you list some shifts or changes that are crucial?
Yes, I think that's a great question. And I think we've gone from, as I mentioned before, sort of saying that exercise should be done and not really thinking about future generations, and mostly thinking mainly about maternal, what the mother is going through. But I think now we can think about, with the DOHAD theory, that in fact, what we see with the mother with regards to physical activity and healthy nutrition, healthy behaviors, we can make a difference in 2 and 3 generations in the future. So I think this is a big shift. And I also think that it's really important that we think about making sure that all of our pregnant clients or individuals that we see are at least walking or doing something to promote physical activity, because I think that threshold that I was telling you about where women or individuals are not getting enough physical activity, I think we need to really watch out for that. And I think the other thing too here is that now that we've got the literature behind us, that we've got the evidence behind us, we can definitely say without a doubt that as long as there are no contraindications to being physically active, for each pregnant individual is different, of course, but if there are no contraindications, then we really need to promote physical activity for health of mother and baby.
Okay, thank you very much. We have a question from Roya. Roya, go ahead. Roya, we cannot hear you. I can see that you have your hand raised, but we cannot hear you. Maybe you are unmuted. I don't know if you are allowed to speak loudly. Maybe you can write the question in the chat. So let's wait for the question, Roya. So write the question and we will answer it later on. Okay, I don't want to have delay in our schedule, but we are waiting for your question, Roya. So now I will ask our second presenter, Professor Laurie Goodyear, to share the screen with us. So hello, Laurie, again. I can see you well, and probably the sound is also well. So just a few words about Laurie. Laurie represents Harvard Medical School and section head and senior investigator. This is the position of Laurie, and also she represents Joslin Diabetes Center in Boston, in US. So I'm very happy that you are here today with us. I know that you are very busy. All professors are busy, but we were talking about that before the webinar, that you are moving from your old facilities to the new facilities, and there are many organizational issues. We are happy that you managed to connect and that you managed to find time for us. And from my side, the last year when we met in Boston, I was very inspired by the research related to the third generation outcomes. So I'm very happy that you are going to share this knowledge with us today. So Laurie, the floor is yours. Go ahead.
Thank you very much, and thanks very much for inviting me to be part of this symposium. Really a great first talk, and I'm looking forward to seeing the rest of the presentation. So today I'm going to talk about some more basic science research, trying to understand how it is that maternal exercise is good. I like to show this figure first because these are actually my offspring, and you can see they all exercise, so that's good. And let's see, there we go. So, one of the real reasons that we started to get into this area was that I remember sitting in a lecture and hearing about these very high rates of obesity in women of so-called childbearing age. And, it's about 37% of women are obese in this age range. And this is a problem, as we just really heard, is because if the mother is obese during her pregnancy, there's an increased risk of obesity in the child. But this also translates to when the offspring becomes an adult with increased risk for obesity and type 2 diabetes. And it doesn't stop there. It goes on to the second generation. So if the mother is obese during pregnancy, there is increased risk for offspring from subsequent generations to develop obesity and type 2 diabetes, fueling this worldwide epidemic of type 2 diabetes, or this, I think, is a big part of what's happening. So is there a means to reduce this risk of transmission? And of course, we have been studying exercise. Now, we'd love to do these types of multi-generational studies with exercise in human subjects, but they would take decades and decades to do. So we have primarily, you'll see at the end, we have done some human studies, but we've primarily used mouse models for this. And the pregnant mother is called a dam in mouse. And our initial hypothesis was that increased physical activity of mothers will improve the metabolic phenotype of offspring. I work in a diabetes center, so of course, metabolism is a major focus. So what am I going to talk about? First, I'll just spend a few minutes on each of these. What's the effects of the maternal exercise on offspring metabolic health? How do the exercise moms actually talk to the placenta and fetal tissues? And then at the end, I'll show a couple slides on what happens when the grandmother exercises. Okay, so this work was started by Kristen Stanford in the lab a number of years ago. She's now a professor at Ohio State working in this area. And we had a couple simple questions. Does maternal exercise affect offspring glucose tolerance? And can the maternal exercise prevent the detrimental effects of a high-fat diet on offspring metabolic health? We already knew that eating a high-fat diet could cause these problems in offspring. So this is the general model that we use. Animals are treated or fed a standard chow diet, and then they're sedentary both in a period before and during gestation, or they're trained by voluntary wheel running, so they do as much exercise as they want. During the breeding period, we don't have them in the cages because we're looking specifically at the effects of mothers exercising, not the fathers. And we often use a high-fat diet to induce this insulin resistance. So do they exercise? Yes, they do. They do a lot of exercise. They do about 2 to 3 kilometers a day during pregnancy. This is less than what they would do if they weren't pregnant, but still you can see a substantial training and training effect. And male breeders are not treated at all. So only the mother is treated, and then the offspring are also not treated. And then we monitor their health through youth out to a year of life, which is middle age for a mouse. And we look at glucose tolerance area under the curve. And just keep in mind that a lower value for glucose tolerance area under the curve is more healthy. So what happens as an animal age? And what if the mom exercised or didn't? So in the white bar, you can see that there is a worsening of glucose tolerance as the animal ages if the mom didn't exercise. But if the mom exercised, you can see that there's a tendency for glucose tolerance to be better. Now, this is in the offspring, right? This is not the mother. So a year of life in middle age, you can see the offspring have improved glucose tolerance compared to if the mom didn't exercise. What happens then if the mom eats a high-fat diet? So here, shown in red, you can see much worse glucose tolerance if the mom ate a high-fat diet. This, again, is the offspring glucose tolerance. But if the mom was exercising while she was eating that high-fat diet, you can see completely normal glucose tolerance in these offspring. I should say there's no difference in food intake or activity levels. So if we look at insulin concentrations, another very important metabolic markers, this also shows the same effect of training. If the mom had trained, the insulins are much better in the offspring. So maternal exercise before and during pregnancy improves the metabolic health of male and female offspring and prevents the deleterious effects of high-fat feeding. So maternal exercise, clearly good in this situation. So we've published multiple papers on this now, as have many others. So I think it's really very well established that in mice and rats, different exercise protocols, this is very effective. So we went on then and we're really understanding the molecular and cellular mechanism that mediate these effects of glucose tolerance. And this work was carried out by a couple postdocs in the lab, Joji Kusayama and Anna Wagner. Joji now has his own research lab. And they did many years of work to really understand what's happening molecularly in the placenta. And what we discovered early on is that really it's an adaptation in the liver of the fetus is what's really happening. So what happens again, if you're not a molecular biologist, don't worry, this will all make some sense towards the end, and you'll see the relevance of this for translational research. But we figured out the whole mechanism by which this happens, this molecule called AMP kinase, and then epigenetic changes in the methylation of different promoters of genes that then improve the gene expression profile in the fetal liver that decrease glucose production, and then we end up with this improved glucose tolerance. So again, years worth of work showing all this molecularly. So then how does this happen, right? We really starting to think, okay, we have to think translationally. Like, is this something we can translate into when humans exercise, and can we develop some treatments to mimic these effects? So the first thing then is we need to really understand, how do the moms talk to the placenta and fetal tissues, or what's the communication that's occurring. So here's this whole cartoon of the schematic of what we know is going on. And so we think about it in a couple ways. What's the maternal placental fetal communication that occurs? And there must be some circulating factors in the mom that is causing these changes in the fetus, right? And wouldn't it be great if we could discover what these circulating factors are? So to do this, what Joji in the lab did was he did a really clever experiment called the serum stimulation experiment, where basically he took serum from sedentary pregnant females and trained pregnant females, and also trained females that were not pregnant. So he takes the serum, and then he takes hepatoblasts, liver cells that were just standard liver cells, and he stimulated, and then he looked at all these signaling pathways and things could this mimic effects? And so what he found was really quite interesting. So this is AMP kinase. This is this molecule over here. It's a key molecule in many different things throughout the body, including this whole regulation of what's happening in the liver. And so if he just treated with serum from sedentary moms, nothing happened to AMP kinase phosphorylation in these cells. But then when he treated with the trained serum, you see this huge activation. But if he treated with a trained serum from a female that wasn't pregnant, so one that doesn't have a placenta, there was no effect at all. So, and then he looked at every aspect of the signaling, and this is what happened. So basically what it's saying is there's something in the trained pregnant serum that's regulating the fetal liver. So serum from the trained dam mimic the effects of exercise in the offspring liver. And only serum from the trained pregnant females activated the signals, leading to epigenetic regulation, suggesting that the placenta is necessary. So you can't just take serum from a woman that exercises and this isn't going to do anything, right? The placenta needs to be there. So then what is this factor, right? Again, thinking translationally, if we can figure out what this factor is, then maybe we can develop something to mimic the way this works. So we did mass spectrometry comparing the serum of a sedentary serum from a sedentary dam versus a trained dam and found 577 proteins. Well, we can't study that many proteins. We need to narrow it down. So 78 of these proteins were shown to be increased by at least 1.5-fold. 19 of these proteins were identified as secretory proteins, which we assume that this was going to have to be a secret protein. And 4 of these proteins were expressed in placenta. Again, we realized that this must be something coming from placenta. And only 1 of these proteins had been reported to phosphorylate AMP kinase, which I showed you before was so critical in what was happening. So what is this protein? Well, it's superoxide dismutase 3, or also known as SOD3, or extracellular SOD. It's got a classical role as an antioxidant, interesting, but also has been suggested to be involved in signaling. So SOD3, we went on and we showed that sure enough, SOD3 protein is increased in the serum of the trained pregnant dams, and that exercise training increases this expression of SOD3, mRNA, and protein in the placenta and the trophoblasts. So we hypothesized then that this SOD3 may be a critical placental protein that plays a fundamental role in fetal development, offspring health, and adulthood. So the next thing we did then was to really test this. We made a placental-specific knockout mouse. You can see it here. You can see the increase with training in SOD3, and then it's completely gone in the knockout. And then we looked at all this effect, and what we see is that we knock out SOD3, we knock out this whole pathway, and especially important is we don't see an improvement in glucose tolerance anymore. So SOD3 is a really important protein. So how does exercise lead to this increase in placental SOD3? Now we have our cartoon over here, and we see the little green dots. That is depicting an increase in this SOD3 protein. So we hypothesized that when a mother exercises, just like when anyone exercises, there's these exercise transducing molecules that are secreted from various factors. So we assume that there's something that's being secreted that then is affecting transcription factors that then lead to this increase in this so important SOD3. And so we did an RNA-seq comparing placenta from sedentary and trained animals, and we found lots of different transcription factors that could be regulating SOD3. But what was really interesting and very highly regulated was VDR, and VDR is the vitamin D receptor. So this was really interesting, right? So vitamin D receptor is increased. And again, thinking translationally, does that mean that vitamin D is necessary for these effects of maternal exercise to improve health? So we could do this in the animals to test this by feeding the animals a low vitamin D diet. But also what we thought about was, could vitamin D treatment function as an exercise mimetic and increase placental SOD3 and improve offspring metabolic health? In other words, can we just treat animals or humans with lots of vitamin D and then we'll have this effect? So we can do this experimentally in the animals. So what happens to glucose tolerance under these conditions? Well, here's our improvement in glucose tolerance. This is just a normal vitamin D diet. This again is in the offspring at a year of age. You can see that with a low vitamin D, you lose this effect. So you have to have vitamin D in order to have this beneficial effects of exercise. In fact, the baseline is even worse. And how about if you give lots of vitamin D? Well, unfortunately, it does not do it. It does not improve glucose tolerance. But if you put it together with training, you actually get a greater effect. So then vitamin D is necessary for maternal exercise to improve offspring metabolic health. Vitamin D treatment alone does not function as an exercise mimetic to improve offspring health. So you have to do the exercise, but you also have to have vitamin D there. So we've done a lot of other work, and I'll just take 30 seconds to show this. But the SOD3 goes up in the moms, and we start to think, well, maybe it's not just having an effect on what's happening in the animal, in the offspring. So we looked at whether SOD3 levels in the serum of the mothers could affect maternal behavior, and we can do this. It's complicated, but we use these heterozygote and knockout animals. You can see they have a lowering. The mom have lowering of SOD3. And so does this affect maternal behavior? And this is in the absence of exercise. This is just by lowering SOD3. So there's a bunch of ways you can measure this in animals. Pups, the number of pups with milk, you can see that's decreased. The number of pups in the nest that the mom makes is decreased. The number of clean pups is decreased. And living pups is also decreased quite dramatically. So SOD3 in the mother's serum is very, very important. So this adds then to our summary that placental SOD3 isn't just regulating offspring health, but it's mediating the maternal nurturing behavior. Okay, so this is all animal studies. What happens in humans? Is SOD3 really this incredible protein that's having these functions? So in collaboration with Christy Adamo, we've done, and the whole group there, we've looked at SOD3 levels. Do they change with exercise? So we compared women that were inactive during their pregnancy or highly active. You can see here. And sure enough, the serum SOD3 levels go up, both in the serum and in the placenta. So maternal exercise increases in both humans and mice. SOD3 and vitamin D are required for these benefits, and SOD3 even regulates maternal behavior. Okay, so in my last minute here, I'm going to just show you this data, really amazing data on grand maternal exercise of F2 offspring done by Anna in the lab. And our hypothesis was that this F2 generation would also be affected by physical activity. So here I'm just going right to the data. Here is the glucose tolerance, just the grandmother was sedentary. And here you can see amazingly that the grand offspring had improved glucose tolerance if the grandmother exercised. If you look at HOMA-IR, a measure of insulin sensitivity, it's also greatly improved in the grandmother exercise. So maternal exercise improves both the health of the first generation and the second generation, and we're working on mechanisms for that. So just finally, there's lots here that's going on. We've got a lot of people working on this. We really want to understand what's happening in the mom that's affecting the placenta. We know vitamin D and SOD3 are important. We don't think it's just metabolism. We think there's effects on the brain and other tissues that we're studying. Of course, the sperm and the oocytes are important, and we're studying that. And we're also looking for the mechanisms of how this occurs with grand maternal exercise. In the humans, what's the role of vitamin D in pregnant women, and what's the optimal maternal exercise paradigm for maximal health benefits? As Michelle talked about, earlier, this is really, really important. So maternal exercise is a means to reduce the transmission of increased risk of metabolic disease. And I think that determining these physiological and epigenetic mechanisms, mediating these benefits of maternal exercise on offspring health, could lead to new recipes to prevent transmission of metabolic disease. Lots of people, this is my group and a couple other groups at Joslin. These are the main people working in this area right now, past members of the lab. Lots of other work we have going on in adipose tissue physiology, human exercise physiology. And Mike Hirschman is our lab manager, who's amazing. And then lots of collaborators that we've worked on through this. And finally, we have funding from a number of organizations. So thank you.
Thank you very much, Lori. Thank you for a great presentation. Do we have any questions? I found your question, guys, so I can see in the chat. So we will keep the questions to Michelle for the end. But maybe just one question to Lori, because we are running with time. So any questions? I cannot see any specific. If you have any, so please write down, and we will try to find time at the end of the webinar. My just summary maybe to your presentation, Lori, that if you want to have healthy grandchildren, it's better to start exercising now, right? So it's not only that you are exercising for yourself, for your baby, but for your grandchildren as well. So we could see from your presentation. And just a quick question about the experiences from the research. Is it difficult to work with the animals to have the third generation assessment? Because, okay, so second generation, it seems quite easy, but the third one, is it okay or difficult? Can you tell us more about this procedure?
Yeah, it's really challenging because it takes a very long time, right? Yes. Because these studies, even the first generation studies, take over a year to do, because we study the animals. They don't have that much of a phenotype when they're young. It's when they're older, just like in humans, right? We don't, most children don't have type 2 diabetes, but so they take a year. And so the second generation experiments take at least a year and a half to do with all the breeding and everything. So it's a very, very long time to do these studies. The reviewer of our paper, one of our papers, actually said they were heroic studies because they take so long to do. But it's very important, right, to really understand that this is not an effect just on your children. It's really an effect that's lasting for a long period of time, for multiple generations.
It's very important data. So thank you for sharing it. And now it's time for Linda. Thank you very much, Lori. So Linda, are you there with us?
Yes, ma'am, I am here.
Okay, that's nice to have. So hello, everyone, and hello, Linda again. I'm happy to announce Professor Linda May from East Carolina University in US. She's the professor and she's involved in obstetric and gynecology, Brody School of Medicine, director Human Performance Lab. And also there is a very nice name, Leroy Walker and Dowager. I don't know what is this, Linda. you have it there, but I have no idea what is the institution. So maybe you will explain it later. But for me, the most important function is that Linda is the chair of pregnancy and postpartum special interest group for American College of Sports Medicine now. So she's co-leading all these events that we are having during the couple months. So Linda, I'm very happy that we are here today and that we managed to do so many important activities related to the promotion of physical activity during pregnancy. So thank you very much for your enthusiasm and also for the high quality research that you are going to present us. So the floor is yours. And explain the name. What was this? Leroy Walker and Dowager. It's really interesting. Okay.
Yes. Thanks so much, Anna, and for everybody for having me here. Really excited, of course, as always, to talk about maternal exercise, and very difficult to follow Michelle and Lori for sure. But I want to follow up with their train and talk a little bit about the influence on the infant cells. So we've talked about 3 generations and epigenetics, and now I want to focus on infant myogenic and adipogenic responses. Let's see if this is going to work for me. There we go. So Objectives, again, I'm going to focus on the mesenchymal stem cells. I'll talk about what that is. I'll look at the undifferentiated data that we have, as well as the myogenic or muscle line and the adipogenic and the fat cell line. All right, and we have already talked about this public health issue. We know that it's a cardiometabolic issue, obesity, diabetes, those types of things related to insulin resistance, neuroinflammation. And ultimately, we know that it's related to fat and regulators from fat, as well as skeletal muscle, but also the mitochondria is what I want to focus on a little bit for my talk. And so how we can use exercise to try to improve this outcome for mitochondria and improve some of these symptoms that we see in the disease state. And so we know that obesity is related to adipocytes, but it's also related to muscle cells. And ultimately, when we look at these cells, it's related to mitochondrial dysfunction. And so there's lots of insulin signaling and inflammatory signaling that the other presenters have already talked about related to that. So I just wanted to remind everybody about the importance of the mitochondria, because we love the mitochondria in the exercise world. So this is my cell and the mitochondria. And you can see off to the top left, it's glucose metabolism. So glucose oxidation, I will be talking about that today. And non-oxidized glucose metabolites, so NOGs, we want less metabolites because we want to increase. So again, less NOGs is better. We want to increase complete glucose oxidation, and I will talk about that moving forward with my slides. And ultimately, you can see that one of these products, lactate, we want less lactate in the blood because that's a sign of a healthier metabolism. And again, the mitochondria would be functioning better. So similarly, if you add insulin to the cells or in our body, we want to have less NOGs. And you can see in the bullet point there, increased NOGs means insulin resistance. So we want less NOGs, less lactate in the blood, for example. But our mitochondria not only are going to use those glucose from carbs, but they're also going to use fats. And so I will talk about fatty acid oxidation as well. So acid-soluble metabolites is another term. Again, less metabolites is always going to be better because that means we're completely oxidizing fat. So we want complete fatty acid oxidation. Some of the other terms I will mention will be palmitate, that's a saturated fatty acid, as well as oleate, which is an unsaturated fatty acid. So let's come back to this idea. We know that obesity is related to prenatal programming and DOHAD. And again, Michelle and Lori have already talked about this. So for me, even though it affects 2 and 3 generations, this is what I wanted to focus on. So for example, as soon as she finds out that she's pregnant, she can't change if she has metabolic syndrome or obesity. And we already know that this is going to influence the placenta, and it's going to influence the baby's development. And importantly, as the other presenters have mentioned, it's going to change the way the baby's organs are developing. And so that's why we want to look at the baby's organs. So then when the child is born, it has an increased risk of now developing that condition because of the maternal environment. And so that's programming. So this example would be the mom programmed the baby to have obesity or metabolic syndrome, for example. But rather than programming towards disease, we like to talk about exercise, as the other presenters did, and how we can program the babies to be healthy. Maybe not super babies, but at least healthy babies. And so with that, I'd like to talk about exercise and really what's the best type, so to speak. So you can see in the picture, is it going to be related to strength or resistance training, or is it going to be related to aerobic training? But we also have other metrics that can be evaluated as well. So there's frequency, there's intensity, there's time, in addition to type, and then volume. And so I'll just briefly mention those, but I'll really focus on type for today. So for my study design, we looked at aerobic only. We also looked at resistance, and then we had a combination group compared to an attention control. And we aimed for the recommended guidelines that Michelle mentioned, 150 minutes moderate intensity exercise. And then fast forward, we have a baby, yay! And then once this baby is born, you can see, yes, we do measurements on the baby and placenta, but we're going to focus on this cord because it has mesenchymal stem cells. So in utero, the baby uses these stem cells to differentiate into other cell types, but we can take those stem cells from the cord and now differentiate them, or we can look at the undifferentiated version, so to speak, the phenotype of the baby at birth. And so we want to use these mesenchymal stem cells to see what's the baby's cellular phenotype. And so my previous postdoc, now Dr. Hjeptovich, was the one who took this bull by the horns and ran with it. And so he started with the undifferentiated cells. So this was thought to be the best phenotype of the baby. These were intact cells that we started to analyze. And again, really looking at this exercise type. Is it cardio, aerobic type of exercise? Is it resistance, or is it both? So what we saw, and this graph is not shown, but basically increased cellular respiration at baseline and max respiration for these MSCs. And again, these babies were exposed to exercise in utero. And then most of the graphs are going to be similar to this one. So you'll see control on the left-hand side, followed by the aerobic, the combination group, and then that resistance or strength group. And then we're going to see the glucose oxidation is improved, especially for that resistance group. So again, we went full oxidation, and then oleate, our monounsaturated fatty acid, you can see it also has increased oxidation as well. So improved glucose and fatty acid oxidation. And again, these are the baby cells at birth, but they were exposed to mom's exercise in utero. And then you can see partitioning is different, and definitely resistance exercise seems to be better. The babies that were exposed to mom's resistance exercise seem to perform better from the cellular level. So then what about when we add insulin? You can see there's this stair-step fashion. Aerobic does a little bit better, combination even better, and then resistance or babies exposed to resistance exercise seem to be the best group even when stimulated with insulin. So very sensitive to insulin. And interestingly enough, remember, we want less lactate in the blood. We can see that what we see in these cellular outcomes of the child exposed to exercise also relates to, at 1 month of age, the baby's lactate in the blood. So less lactate was associated with improved cellular outcomes as well. And this also tracked with what we saw not only in the cellular outcomes that you can see in the x-axis, but again, at 1 month of age, also the body fat percent. So those cellular outcomes track with the whole body outcomes at 1 month of age. So we continued to analyze this and look at basal and insulin stimulated. As I said previously, there's an improvement in the insulin signaling and the MSC respiration. Not only glucose, but fatty acid oxidation is improved in these cells from babies exposed to mom's exercise in utero. But as Lori mentioned, there's lots of cell signals that are associated with the mitochondria and the cell respirations. And you can see AMPK, for example, was already mentioned. Citrate synthase, for example, was upregulated in these cells as well. So definitely a sign that these mitochondria in the cells are functioning better because of that exposure to exercise. And then when we translate this to the whole body outcomes, again, you can still see that when we look at the exercise exposed babies to the babies not exposed to exercise, we can see that the babies at 1 month of age have a lower BMI score. And this is specifically associated with decreased body fat and increased lean mass. So again, what we see in the cells translates to these whole body outcomes for the child, and we can still see these outcomes. Again, cellular respiration also tracks with the 6-month body fat as well. So improved cell respiration at birth for those undifferentiated phenotype, and improved or lower 6-month body fat and 1-month body fat. All right, so those were findings from the undifferentiated cells. Some people think that's the best way to look at the baby's phenotype from a cellular perspective, but similar to what the baby does in the dish, we can also differentiate these cells into a myogenic or muscle line, and then a adipogenic or fat line. So let's look at the muscle or myogenic first. So regardless of pre-pregnancy BMI, what we found for these muscle-like cells of the baby was that we see something really similar. So again, increased glucose oxidation and very sensitive to insulin. And similarly, as what we saw before, we also see that the improvements at the cellular perspective is related to that 1 month decreased blood lactate. So again, the cell and the whole body outcomes match, again, suggesting the child might have a decreased obesity risk, again, regardless of whether the mom was obese or not. Previously, the prenatal programming said if she's obese, baby's programmed to be obese. But with this exercise, we can change that programming. So again, for the myogenic, still controlling for pre-pregnancy BMI, exercise type, fetal sex, breastfeeding as much as we can. And you can see that we still have similar results in the myogenic or muscle differentiated cells compared to the undifferentiated, where we have the stair-step fashion, aerobic to combination, resistance being the best. But all of them seem to be insulin sensitive, but especially that resistance exposed group is very insulin sensitive. And again, when we look at the insulin stimulated, so are they insulin sensitive and how does that relate to the whole body outcomes? You can see that improved outcomes, improved insulin resistance at the cell seems to be related to that 1 month decreased body fat of the child as well. And at 6 months of age, it's still related to their lower body fat stores as well, related to the muscle cell line. And then next, since there seems to be an insulin sensitivity, we wanted to analyze these muscle cells, the ones that were insulin sensitive versus not insulin sensitive. And so you can see that we do see differences there in the cell line of these muscle cells from the babies exposed to exercise. So low sensitivity, high sensitivity were the ones exposed to exercise, for example. And you can see that this is also related to not just their weight, but less body fat at 1 month of age. And they are definitely showing signs of metabolic flexibility. They're able to lose glucose and fats equally well. And so that's what this picture is showing as well. So the low insulin sensitivity to the high insulin sensitivity definitely has improvements in completely oxidizing fats. And as you would imagine, the improvement in fatty acid oxidation of those myogenic cells is also related to lower body fat stores at 1 month of age, as well as 6 months of age. So again, still suggesting even that muscle line is suggesting that decreased obesity risk as well. So that was the undifferentiated, definitely see improvements in that phenotype, but also in the myogenic phenotype. So then the last one from a metabolic perspective, we have muscle, and then on the other metabolic perspective is going to be the fat cells. So what happens in the fat cells of these babies exposed to mom's exercise? So this was Dr. Claiborne that is leading these studies. And what you can see in the top right, oil red O is a stain for lipid droplets. And you can see that the exercise exposed babies have less lipid droplets in these cells that were differentiated into the fat cell line compared to the control babies. And not only that, we can see that the decreased lipids in the cells is again associated with that decreased body fat at the whole body level as well. But what about exercise type? So again, we can see that the control babies are over here on the left-hand side, and then any type of exercise is beneficial, but definitely when she does that resistance or strength training alone or in combination, the babies are going to have even less lipids in their differentiated fat cells. So they seem to be a little bit better as well. And this is very exciting when we talk about other exercise metrics, not just the exercise type, but what about how much she does each week? And we see for these fat cells in particular that the amount of exercise that she does each week is associated with those lipid droplets. So if she does more exercise on a weekly basis, that's associated with less lipid droplets in those fat cells. And same thing or similar, we can see more exercise each week is associated with less body fat. So again, the cell and the whole body outcomes match, but it's not just an exercise type, but it's how much she's doing each week is important, with the recommended guidelines being right here at 150 minutes. All right, so in summary, what we see is that maternal exercise dose and type are important. We see improvements in that infant myogenic phenotype, as well as improvements in the infant fat phenotype. And this is really important. We see that there's an association not just with those cellular phenotypes, whether it's undifferentiated myogenic or adipogenic, but this relates to the whole body outcomes. So what we see in the muscle-like cells and the fat-like cells is related to decreased body fat on the whole body level and increased lean mass for the children at 1 and 6 months of age. So definitely showing signs that we are hopefully decreasing the child obesity risk. So lots of people to acknowledge, hopefully I haven't left anybody off, but also want to acknowledge the funders, American Heart Association, as well as NIH and the Thrasher Foundation, as well as Brooks. Brooks Shoes provides or Brooks Incorporated provides free shoes to our pregnant women, and Fleet Feet is our local provider. And then not only do I have tons of collaborators, international and across the US, but we have lots of people on the study team, boots on the ground that really get things up and running for us. So thank you so much for your time, and let me know if you have any questions.
Thank you very much, Linda, for the third great presentation. So amazing outcomes, amazing data that you are sharing with us, especially, this information about the obesity prevention that we are fighting all around the world. And it's really, really important to have this information. So guys, do you have any questions to Linda? I can see that already a question to Lori was written. So let's wait to respond for the end of the webinar, but maybe to Linda, one question or two questions, short questions to go ahead with. Not yet.
I have a question. I can ask a question.
Okay. Okay, of course, Lori.
So the resistance exercise data it's just so interesting. So I'm just wondering what you think the mechanism is for the resistance exercise having these more beneficial effects, greater effects.
Wow, that is an excellent question for sure, like the $1 million question. We don't really know, but we do know that there's different molecules, and I believe you and Chrissy Adamo have looked at that, the myokines. And so I'm imagining that there's going to be different myokines that are released when we're activating the skeletal muscle in a resistance strength type of way with intermittent exercise versus a cardiovascular type of aerobic exercise. So maybe it's myokine related. And funny enough, one of the things that we've also started to look at is vitamin D. And so I appreciate that you mentioned vitamin D. And we don't know if vitamin D, because it's also related to the anti-inflammatory pathways, is released differently or managed differently in resistance training versus aerobic type of training. So there's a couple things that we're looking at in addition to the vitamins and possibly myokines and other growth factors. There's specific growth factors that are going to be released from the muscle that might be related to this as well that we're also tracking down. So a few things, but we don't know yet. Still doing some studies. Great question.
Yeah, it'll be interesting to also just to look at the placenta itself, right? And see if there's a difference in the placenta with the resistance versus endurance type exercise. But really excited.
Absolutely. Yes.
Absolutely.
We're just starting to look at that with some of the lipids and the growth factors, but we don't have any data to share on that. But absolutely, definitely the big black box that we're hoping to get some answers for sure. Appreciate that.
Okay, thank you very much. I can see two more questions, but Linda, let's wait for the end and I will go ahead with my presentation now. Okay, so now I'm sharing the screen again. Now I'm sharing the screen again to present the outcomes of the HIT Mama project. Okay, just confirm that you can see my presentation, please. Yeah. All right, so let me move it up. Okay, so just a short introduction. Let me move it.
Anna, we don't see your presentation mode.
Yeah, now it's
We
presentation
see
mode.
Yes.
Yeah. Okay.
Thank
you very much. So as I mentioned, I represent Gdańsk University of Physical Education and Sport in Poland. So this project was run at my university, and it was inspired by the lack of data on higher intensity exercise. So usually we have information how continuous training can impact maternal health. A little bit we know how it can impact fetal health, but not a lot is available on higher intensity exercise. I mean around 80% or 85 or 90% of heart rate max. And especially I and my team, we were interested in high intensity interval training. Of course, we have a lot of different modalities of high intensity interval training. It can be vigorous intensity like 70-90%, and also much higher, more than 90% of heart rate max. But we were interested if we can perform such mode of exercise in pregnancy, because there were many myths related to that and many concerns that this is too intensive, that we can, take too much blood and the baby is not properly nutrient. And this is the project that we are running. It is 10-year project, and we are now on the phase that we are analyzing the maternal blood, maternal urine after 8-week cycle, and of course we are going to continue this analysis in infants, and also during the next stage we are going to take the samples from placentas. Study participants, we had 2 groups, high-intensity interval training group and educational group, to compare the outcomes. And the inclusion criteria were first normal glycemia, normal lipidemia, normal tension, and normal course of pregnancy. And importantly, during this 8 weeks of exercise, all these parameters were maintained in norm. This is the protocol of the study. So women came to the laboratory, they had the blood and urine collection, and after the same meal, they had some functional parameters assessed. We had the exercise test up to refusal around 2.5 hours, and this was a standard test, but importantly, it was up to refusal, which is usually not performed during pregnancy. So we have the outcomes, we have the parameters collected from the blood and from urine immediately after this up to refusal exercise. And then the third time point was around 60 minutes after the test. And we monitored the intensity. So based on the CPET, we set the heart rate for anaerobic threshold, and it was around 87 ±5% of heart rate max. And all sessions were recorded with heart rate monitors. And you can see the red color. So this is the intensity above anaerobic threshold. So not a lot. It was a few minutes during each session, short boosts of high intensity exercise, but still it was something which was not generally recommended on the time, and it is not usually recommended, and sometimes people are afraid that it's too intensive. And starting from the very frequently questioned, no adverse effects of this intervention were observed, neither in the obstetric nor neonatal outcomes. And we wanted to analyze what was going on in the maternal body. So as I mentioned, we don't have outcomes yet related to infant development or placenta development, but still we wanted to observe what was going on in terms of exerkines, for example. And one of the myokine that we were analyzing was irisin. Irisin is a cytokine which is important for trophoblast cell differentiation. So that means that if there is too little, that the level of irisin is too low, there is a higher risk of some problems with growth of the baby. And when we have too high level of irisin, there is a higher chance, high risk of having macrosomic babies. So the babies are too big. So somewhere there is an optimal level of irisin, and we were trying to assess how we can help to achieve this optimal level of irisin through exercise. And some authors observed that usually irisin increases markedly during pregnancy, and it is expected to have higher irisin level because when the level is reduced, the condition is related with increased ratio of preeclampsia or gestational diabetes mellitus. And what we found, and what is generally known, that physical exercise can impact the level of irisin, and then irisin can impact all the pathways related to, for example, inflammation or adipogenesis. And also it is related to insulin sensitivity. It can help with gestational diabetes prevention. But on the contradiction to other authors, we haven't found increases of irisin through exercise. Importantly, we found that it is correlated with glucose level, And the more irisin was produced, the better glucose level, the lower glucose level, and lower outcomes related to homeostasis of glucose. But also what we wanted to check was if after one session, if it's possible to have better outcomes related to irisin production. And in fact, when the production of irisin was more effective, the lower cholesterol level and lower LDL cholesterol was found. So that means that these processes were related to favorable lipid profile. And we haven't found this correlation. Actually, we found completely different correlation in the control group, so in women who were not exercising. So we could conclude from that there is an optimal level to have the production of irisin induced by exercise. And as I said, there is a slightly decreased between the second and the third trimester when we have healthy, physically active participants. So our conclusion was that they don't need so much irisin to regulate all the processes. And in the next study, we also confirmed the same outcome. So when we had the high intensity interval training, we could observe that there were increases and decreases in irisin levels, and it was very important for us that most women experienced decreases in irisin, and there were slight decreases, so it was not a very substantial change, but still we couldn't expect that the irisin will be higher at the end of pregnancy. And another biomarker that was observed, it was insulin. And it was surprising that for insulin, we observed more increases than decreases, and it was a substantial change for high intensity interval training group. So we should suspect that when you exercise, the insulin level should be a little bit lower, because insulin production can be an effect of impaired glucose uptake and utilization. But as I mentioned, we haven't observed any problems with glucose level, which was confirmed by other blood assessment. So we could assume that insulin stimulates muscle blood flow and insulin signaling in skeletal muscle, and that the effect of the increase in insulin was related to training adaptation, typical for the energy demand for the HIIT session. And we could also confirm this outcome by other analysis using the bioimpedance, where we observed that the free fat mass, the percent of free fat mass, was substantially improved in the high intensity interval training group. So only in this group we could observe that outcome. And another hypothesis was that maybe the increased insulin level was related to increased glucose transport to the fetus, caused by greater efficiency of the fetal-placental blood exchange and better glucose utilization by the fetus due to intense maternal exercise. And of course, other authors have some outcomes that can support our thesis. So from that study, serum from pregnant women could contain factors that increase glucose transporter 1, protein expression in vitro, and physical activity appears to differentiately impact key placental transporters involved in the transfer and availability of nutrients from mother to fetus. So maybe that was the reason why we could observe higher levels of insulin. And our study was confirming this thesis. We were assessing placental growth factor, and it is a biomarker of preeclampsia and biomarker of some disturbances in some disorders related to growth of the fetus. And we presented this study, which was quite interesting, that we enhanced the production of PLGF through HIIT. It was not statistically significant, but still we could observe the trend that we can support the production of PLGF. So probably the production and the stimulation of proper development of placenta through heat. And another biomarker, leptin, again, we could observe increases in that marker. It was not statistically significant, but still it was. And we thought that probably there are some concerns. So are these women more likely to have increased fat storage, or there are some problems with the energy metabolism? But again, I'm coming back to the study about the body composition. We observed lower percentage of fat mass in the heat group. So that means that we shouldn't be worried about this lipogenesis. And also, we must remember that leptin enhances insulin sensitivity and increase glucose uptake and oxidation in skeletal muscle. So maybe this function was enhanced through heat. And another important information, leptin is involved in reproductive functions, and maybe through heat we stimulated reproductive functions of leptin during pregnancy. So it is important, starting from implantation through trophoblast differentiations and also the angiogenesis in placenta and fetal development. And the last one was really, really surprising. We observed completely opposite trends in the heat group and in the edu group. We observed that the resistin increased, substantially increased, and the resistin substantially decreased in the educational group. So different outcomes. And I didn't say that the educational group was the comparative group. The women came to our laboratory, they had the same assessment, but they didn't have the intervention of exercise. So we were discussing, we had lectures, so therefore it is educational groups, but they just were recommended to perform a physical activity of moderate to vigorous intensity by their own. So should we worry about resistin? We know that resistin impairs glucose uptake by adipocytes, increases plasma glucose concentration, and thus decreases insulin sensitivity. But as I mentioned, we haven't observed these problems in our group. So both normal lipidemia, normal glycemia were maintained throughout the study. And again, we found other papers related to the connections of resistins, and resistin modulates glucose uptake and glucose transporter 1. So it was the study and the author found that resistin can cause activation of pathway related to GLUT1 synthesis, and this treatment resulted in increase of placental glucose uptake. So what about the kidney function during high intensity interval training and urine analysis? It is more and more attention paid to renal function and also the kidney diseases. We wanted to check if having very intensive exercise, we are not putting women under some risk related to hypoperfusion. Some outcomes were completely typical changes. Some were a little bit unexpected, but first of all, we didn't find any negative impact on markers of renal hyperperfusion after very intensive exercise. And also we could observe that maybe there are some pathways enhanced for fighting with albuminuria and also with preeclampsia. So another study related to analysis of urine proteome. It was only 9 participants in the heat group and 9 participants in the edu group. It was a pilot assessment, but we used this proteomic analysis with the SWOTCH-MS method. And interestingly, before the 8-week intervention, we found it was 336 proteins which were substantially changed, and we found only 43 which were substantially changed after 8 weeks of heat. So that means that there is some adaptation to exercise, to high intensity exercise, which can be analyzed in proteome, urine proteome. And before 8-week HIT program, interestingly, we found a lot of responses related to neutrophil degranulation. So the process is related to immune system. The yellow color you can observe here, the yellow color, it's in this nice nodes. And immune system was mostly impacted before 8-week high intensity program. And after 8-week high-intensity interval training program, we could observe changes mostly in cellular components, and especially it was interesting that MIF was mostly impacted before and after the training, 15 minutes after very intensive exercise. So MIF is important for immune and inflammatory response, and it is also involved in the development of the embryo and placenta because it is the mediator of angiogenesis and vasculogenesis. And also it is related to increase in SFLT1, so a very nice, a very important protein related to some risk related to adverse perinatal outcome, and it is usually a biomarker, especially in relation to PLGF. These are the biomarkers suggested for preeclampsia. So what we wanted to say that MIF is related to increased FLT1. So maybe this is the explanation how we can improve, how we can prevent preeclampsia through intensive exercise. And the last one was after 60 minutes after CPT, and it was after 8-week program. You can see on the graph gray color, the ring gray color means that these proteins were changed only after 8-week HIT program. So that means that we could observe some new responses to exercise, which was not observed at the beginning in the group of these 330-something proteins. So our conclusions were that during physical activity during pregnancy, regardless of intensity, optimizes pregnancy and fetal development, because we know that we can prevent different disorders related to gestational diabetes or preeclampsia through moderate intensity exercise. But here we could say that also intensive interval training can be recommended for normal pregnancies. And also through the interconnection of a variety of hormones, growth factors, and cytokines regulating all processes in maternal body, we can say that we can avoid different pregnancy disorders. So this is one thing, but potentially we create better conditions for fetal development. We don't know these processes very well yet. So this is our first analysis, and we hope to analyze it in the future. We would like to invite all researchers who would like to help us with that. These are the babies from our project. So they are healthy babies, happy babies, and this is the research team. Of course, not all people were mentioned here, but this is our team. So I will stop with the presentation about the high intensity interval training here. If you have any questions, I will be more than happy to respond at the end, but give me 2-3 minutes for the Global Active Mother Day to invite you for our initiative. So as you could see, all the presenters, Michelle, Lori, Linda, they devoted their life to different analysis, and we would like to share this knowledge with you during different events. And one of the events is the Global Active Mother Day. So this is the idea that we have education, we have exercise classes, but everything is evidence-based. And this is a concept of Be Active Day, which is run in Europe for many years already, and we would like to repeat it. So we have several organizers from different countries. So thank you very much for that. You can find them using the QR code. So different, I can say, instructors, trainers are doing something. So it can be education, it can be exercise classes, it can be health services. We have also online offer for you to share for your clients, for pregnant mother, for postpartum mother, for all families. And also we have nice materials, video materials. This is an example. We have more, but we didn't have time to post everything. But yes, the videos also are coming. So if you would like to join us, just screen this QR code and fill in the questionnaire and just let us know what do you want to do. So I know that the Global Active Mother Day is in 3 days, so not a lot time to organize something. So maybe you can just go and support your colleagues, but maybe it will be good to have this, I don't know, the note in your calendar that in the next year, we will meet on 11th of May and we will do more events. We will organize more education, evidence-based education for our pregnant women. And I will also share some information about our networking. So we have session in Atlanta, which will be on May 29 for ACSM members. We have also the special interest group for physical activity in pregnancy under the umbrella of European College of Sports Science, and we will meet on Thursday as well, but it will be the 3rd of July, and a very nice session run by Professor Linda May in Rimini. And also, I repeat the information which was presented by Michelle about the pre-conference of Canadian Society of Exercise Physiology, which will be held in October 14th, 15th. Okay, so if you would like to find more information, this is our website where you can find the recordings, the information how to organize something, and we will be more than happy to have you with us. All right, so I'm stopping sharing my screen. Any questions? Any questions to new questions to the research? I will go to the beginning. Let me go to Michelle first. It was a question about today Canadian conference about Michelle Motola. I'm not sure if this was posted already, Michelle. I think that we will post the information on our website as well, all right? So it will be easier for other people to find. So it will be done. And also, what has been the real motivation for Canadian ladies to be more active? It was to Michelle as well.
Yeah, that's a great question. I think Canadian pregnant individuals are just as inactive as everybody else around the world. So I think it's up to us as educators and fitness professionals and researchers to get our information out there to help promote physical activity for pregnancy. And we're now seeing that the information, the evidence is overwhelming, as you saw today from our speakers, that it's very important that we get the message out and that we tell pregnant individuals, pregnant clients, however we can, that daily physical activity is extremely important for the health of themselves and also their future offspring. So I think it's really important that we get that message out.
Okay, thank you very much. And another question to you, what has been the real, also that one, because you have one of the most successful models and the implementation to me is way harder to set a guide. Okay, so I think that was a comment, so that you are very successful, Michelle. And another question, since you have one of the most successful models, and I want to know what really helped, because to me implementation is harder than just setting the guideline. Okay, so I think that you already responded to that one. Mm-hmm. Yeah, but there was also another question related to, are there any researchers we should follow that are going to work on the upper limit for PA apart from Dr. Davenport? So it was also, Michelle, to you.
Yes, I think Anna's one of the leading investigators looking at that as well, because she is, Anna, you've done a nice presentation there on the HIT. But I do think that there's a number of individuals around the world. I can think of the group from Norway, So
Lene Hagstad, Lene
yes,
Hagstad, she's doing a lot of research on
doing
the
that,
really high intensity, because mine is like, around 85, sometimes 90, but generally we are keeping the anaerobic threshold, but she's doing 90% of heart rate max, Yeah,
and I think a lot of the information right now is just looking at acute exercise, whereas I think we need a lot more individuals out there in the field that are going to be looking at the chronic effects of high intensity exercise. And again, I think we have, because we're pushing the limits, we need to be very cautious about this. So it has to be a coordination between the medical professionals and the athletes. And I'm talking about high level athletes now, and I think we need to be very cautious about pushing the envelope, but we're getting there. But I think it's something very important for the future.
Yeah, I totally agree. And more athletes would like to combine maternity with their normal regime for sport performance, and that's really difficult sometimes, right, to combine everything. Yeah, so we need more research on that. Okay, thank you very much, Michelle. And now the question to Lori. Hello, this is a question for Lori. Great presentation. I have a few questions about methods, but I ask the one I have about vitamin D. Vitamin D is connected to bone health and thyroid function. Have you considered this connection? Also in human, vitamin D is made in the skin through using the sun. Is there a connection between sunny mouth and COD3 levels? Thank you again.
Yeah, these are very interesting questions. So in terms of the bone health and thyroid, we have not investigated that, but I think it's probably very important given this connection with vitamin D. And certainly it could be a factor in what's happening when the moms exercise, that there could be adaptations in the bone or the thyroid that are then speaking essentially to the fetus. And then in terms of the vitamin D, when we first made this discovery, we were very interested, and we still are very interested in understanding this because of obvious seasonal variation. And we haven't quite figured out how to look at that. We tried in a cohort that we were investigating with a group in Denmark, trying to see if we could look at the time of the pregnancy, but the pregnancy lasts for a period of time, and then the SOD3 we think are higher in the second trimester. So we really, the study wasn't designed for that purpose, but it would be very important to try to understand if SOD3 levels are different at different times of the year and depending on where the phase of the pregnancy, because that could obviously, if this carries through to humans, then this could affect the outcomes for the offspring.
Okay, thank you very much. And I'm not sure if this is to Lori or to Linda, or to four of us. What is the effect of physical exercise on the, I'm not sure if I'm pronouncing it correctly, Paekersin activity of MACCs? So I don't know this biomarker very well, but maybe some of you can.
Yeah, so the paracrine activity is just a feedback activity to the MSCs, and I responded to, I think that was Michael's question, or Michael.
Yes,
it's a great
Professor
question.
Michał,
I don't know. Yeah, we haven't looked at the paracrine activity of the MSCs. We're just starting to do a few more studies related to what they're releasing and response to what we give them. So yeah, great question. We don't know yet.
Okay. So we have some new subject to research, Linda. Yes. Okay. And there's a comment, not a question per se, but rather a comment. It's exciting to see all the research that still continues to come out from the Active Mom study, Dr. May. So congratulations to Linda. It's nice to hear that. Another comment. Huge thanks for all your amazing work that supports us on the ground and in our research and there is Dance Mama organization in the UK. We will take our posts over the periods period with the Global Active Mother Day. So thank you very much for your engagement in the Global Active Mother Day. And actually, Michelle, it was your idea, I must say. It was your idea it was from Denver or from Boston. I don't remember, but it was your idea to have the Global Mother Day. I wanted to have it in September, to have the Global Day for Physical Activity during pregnancy, but to say why not to do it in May, because it's the Mother Day on the 11th of May. So it was. And I hope that we will continue. Can you please put your questionnaire link in the chat when you are able to? So I'm not sure if I can use the link now, but if you go to our website, everything is posted there. So if you would like to join us, there is the link and everything that you can use. And also, let me check. It's a comment. I cannot see it. I don't know. Oh, so you already responded, Linda, to that one. Congratulations, great presentation. Thank you. Thank you. Much more to do in dance. Thank you. All great presentations. So these are the comments at the end. So, ladies, it was really a great pleasure to have you today. So before we finish that webinar, I will share very quickly my screen again. Of course, I would like to thank you very much, Michelle, Lori, and Linda, for your time. Again, I know that you are busy, but you found time, and it's really appreciated. And we are sharing this knowledge wherever we can. And also, what we would like to do, I would like to thank a lot our organizers, so Małgorzata Perl, Małgorzata Prochownik, and Dagmar Adams. Without you, I couldn't do this webinar. So thousands of emails, thousands of posting, and I really appreciate your time and your devotion. And I would like to invite everyone to cooperation with us. This is my email address, but also there is the general InfoNepe email, so you can send your questions if you have more questions about this webinar, but also you would like to collaborate. I would be more than happy to respond or find someone who can respond to your questions. So, dear presenters, would you like to have a comment on that yet before we finish? Lori, Linda, Michelle? No? Nothing to add. So guys and participants, thank you very much for being with us. And I hope that you will use the recording, which will be posted on our website. So thank you once again.
Thank you,
Thanks,
Anna.
Anna. Thanks, everybody.
Thank you, everybody. Bye-bye.
Thank you.
Bye-bye.